The next topic that I want to discuss in terms of where genome engineering has had and can have a big impact is still on this theme of different kinds of functional groups in biology, in living cells, and ways of broadening what cells can do. So I'm going to go through this section and really the rest of these slides faster. I probably won't be ending today, meaning I probably won't finish all of this stuff today. But I want to cover this because a lot of these topics have more to do with protein engineering than metabolic engineering. But in the earlier portion of our course, we've talked about the many intersections between protein engineering and metabolic engineering. So here we're going to switch gears slightly to thinking less about metabolites and more about maybe amino acids and proteins. But we're keeping the same idea that the cell might be doing things that can compete with what we want to do, or at least the introduction of more chemistry. And so if we revisit the codon table, for example, which we've spent time on before, we've talked about the degeneracy here, the idea that an amino acid is encoded by multiple codons. In this case, for example, serine, you've got 4 here and another 2 there. Even the termination of translation is encoded 3 different ways, 3 different codons, and you don't need that. There was an effort in the lab where I was a postdoc, but well before I got there, this was actually happening in parallel to the work that I discussed on engineering aldehyde biosynthesis. You had the technology of MAGE, the multiplex genome engineering, being used to recode genomes. And this recoding meant taking this TAG stop codon and trying to eliminate it from the entire genome, and for that matter, succeeding. And some reasons why you might want to do that is because, in general, if you have a different code here in your organism than exists in nature, since this codon table and the genetic code is almost entirely conserved across all different forms of life, then when you have things, say, like viruses that rely on the host machinery to perform translation to make their proteins that give them the ability to replicate, etc., they would be confused. They would not make either the right protein, or they wouldn't terminate translation where they need to, thus giving you increased odds of virus resistance. Really depends on how much recoding you do. More close to home, and to this idea of cellular chemistry, if you have a codon that's freed from the genome, you can rededicate it. You can hijack it, reassign it to a new amino acid, to a non-standard amino acid, which we consider an amino acid that isn't one of the 20 natural building blocks of protein. And something I won't talk about, but also interesting, is one reason why you might want to do that is because then, well, especially coupled with this idea of virus resistance, if you have this strain that's really different and maybe poses some advantages over wild type organisms, then you might want to contain it genetically, which you can do by making it dependent on supplying some kind of non-standard amino acid. So just to be able to briefly understand how this works, going back to like a more basic concept in molecular biology, aminoacyl tRNA synthetases are the enzymes that attach transfer RNA, tRNA, to an amino acid. And that's what gets sent to the ribosome in order to get matched with some kind of mRNA at the right codon-anticodon pairing, and then add it, that causes the transfer of your amino acid to the growing peptide chain. So you can put in non-standard amino acids into proteins or incorporate them the same way. You just have a different tRNA. So it has an anticodon here that corresponds, in this case, to that stop codon. And then you supply your non-standard amino acid. This is just an elongation factor. Here you got your mRNA, so where you have this UAG, you would have your non-standard amino acid come in, and it goes into that spot in the protein. So that's not really too important for what we're going to talk about today, but it's just to give you a backdrop for why this stop codon, which is also the least frequently occurring stop codon in E. coli, is the one that's of interest. These systems were developed well before there were any recoded genomes. And this technology was really powerful for being able to do all kinds of interesting protein engineering. So from the genome engineering standpoint, since that's the focus of this lecture, the way that one got to a recoded E. coli strain involved this kind of procedure from the Church lab. So ordinarily, you have 2 release factors in your E. coli strains, and they are proteins that mediate this recognition that this stop codon is trying to terminate translation, or is that it encodes the termination of translation. So you know, your release factor 1 or 2 would come depending on what the stop codon is. If it's a UAG, it's exclusively recognized by release factor 1. And so if you had a way to change the UAG to do what's called synonymous substitution and make it a UAA or a UGA, then you could presumably get rid of RF1, and RF2 would just take care of the rest. And so in fact, that's what they did in this work, which took many years. Excuse me. Okay, so all native UAG codons were recoded to UAA, and then you had so that essentially so that meant that there were no UAGs around, but you still had this release factor, and so if you were to ever put back in a UAG in a different part of the genome, it would still terminate translation. But then you could delete the release factor, and now you've made UAG have no meaning at all, and it's not in the genome. So then you could put back UAGs into the genome, restoring them not at their native loci, but within genes, within protein coding sequences where you would want a non-standard amino acid to go. And so that, since there are 321 or 314 UAG codons within the genome, you have too many to be able to do this multiplex genome engineering all in one strain. And so there was a development of some clever techniques, including this hierarchical assembly framework known as CAGE, that uses conjugation to be able to exchange genome segments among different bacterial strains. So if you could essentially combine through conjugation different sections of a genome and do that in some way that you could select for, then in parallel you could essentially divide the genome into different chunks, and in parallel you could make your substitutions. And if you had these 32 different regions, you could combine them in pairs. And so then you'd end up with a strain that had segments 1 and 2 combined, and so forth. And then you could keep doing this pairwise assembly until you got a fully recoded genome, where then you could knock out the release factor 1 encoding gene.